Welcome to the deep dive. Today, we're exploring something really quite remarkable: bioluminescence. That's the ability of living things to produce their own light.
That's right. And we're going to focus in particular on perhaps the most familiar example, fireflies.
Ah, yes, that summer evening glow. It feels almost magical, doesn't it?
It does. But today, we want to go beyond just, appreciating the sight. We'll dig into the how and the why behind it.
Okay.
We're drawing mainly on the key points document and a survey note analyzing firefly bioluminescence. Our mission really is to shed some light, pardon the pun, on just how complex and impressive this natural process actually is.
Excellent. So let's start with the basics then. Bioluminescence. What exactly is it?
Okay, so bioluminescence is fundamentally light that's produced by a chemical reaction inside a living organism.
Right. How's that different from, say, fluorescence?
Good question. Fluorescence is different. That's when an organism absorbs light from an outside source, like UV light, and then immediately emits it back out. Think of some corals.
Okay. Absorb, then emit.
Exactly. And then there's phosphorescence, which is similar, but the light emission is delayed. It keeps glowing for a bit after the initial light source is gone, like glow-in-the-dark stickers.
Got it. So the key thing with bioluminescence is it makes its own light. It doesn't need outside light to start it off.
Precisely. It's all driven by internal chemistry. That makes it unique and actually incredibly energy efficient. We'll definitely come back to that point.
Interesting. So fireflies, then, they're probably the first thing people think of. Why do they light up? What's the point?
Well, research points to a few main reasons. The biggest one seems to be for mating.
Finding a partner.
Exactly. Male fireflies flash in specific patterns to attract females of their own species. And what's fascinating is that these patterns are unique to each species.
Wow, like a secret code.
Kind of, yeah.
Yeah.
A female will generally only respond if a male flashes the right pattern for her species. You see this with different groups, like Photinus or Photurus. They all have their own distinct light-based courtship language.
What?
And the females. They often just sit tight, maybe on the ground or low vegetation, and flash back with their own specific timing. So it's this quite sophisticated dialogue happening in flashes of light.
A conversation in light signals? That's incredible.
It really is. But finding a mate isn't the only reason.
Oh, what else?
Well, especially in the larvae, but also some adults, the glow acts as a warning signal to predators. It's called aposematism,
A warning, meaning?
meaning don't eat me, I taste bad.
Yeah.
Or worse.
Hmm.
They produce these chemicals, lucifugins, for example, that make them unpalatable, even toxic. So the light basically advertises that defense. It's like they're saying, look, I might seem like an easy snack, but trust me, you'll regret it.
A very effective visual warning, then. Any other functions besides mating and defense?
Well, there seems to be some general communication within the species too, and then there's a darker side.
Oh.
Some species, like certain Photurus fireflies, are actually predators. They mimic the flash patterns of other species.
No way. Why?
To lure males of those other species closer, not for mating, but to eat them.
Whoa, that's unexpected. Deception by light.
Exactly. And sometimes they might even do it to steal the defensive chemicals from the prey they eat. Nature can be quite cunning.
Clearly. Okay, so this light show. Where on the firefly does it actually happen? Is there a specific part?
Yes, there is. It's produced in a specialized organ called the lantern. It's usually located on the underside of the lower abdomen.
The lantern, okay.
And it's quite a sophisticated structure. It contains special cells called photocytes, which are absolutely packed with everything needed for the light-producing reaction. Behind these cells, there are layers that act like reflectors, bouncing the light outwards, making it more visible. And crucially, the lantern is connected to the firefly's breathing system, the tracheal system,
Why is that important?
because that system delivers oxygen, and oxygen is a key ingredient for the chemical reaction that makes the light.
Ah, okay. So the lantern is the factory. Let's talk about that reaction. What are the key ingredients, the molecules involved?
Right, the chemistry is the heart of it. You need a molecule called luciferin,
Luciferin.
an enzyme called luciferase.
Luciferase.
Sounds similar. They work together. Then you need oxygen, which we just mentioned. You also need ATP, that's adenosine triphosphate, the cell's main energy currency.
Right, ATP. I know that one.
And magnesium ions are important too.
So luciferin and luciferase, what do they each do?
Okay, think of luciferase as the catalyst. It's the enzyme, the worker molecule, that makes the reaction happen quickly and efficiently.
Got it. The facilitator.
Exactly. And luciferin is the substance, the molecule that actually gets chemically changed, oxidized, to release the light.
Okay, so how does it work? Step by step.
Basically, luciferase helps luciferin react with ATP using magnesium. This creates an intermediate compound. Then this intermediate reacts with oxygen. That forms another very high-energy, unstable molecule in dioxytannin.
Dioxytannin, okay.
Because it's unstable, it quickly breaks down, and when it does, it releases oxyluciferin, carbon dioxide, and light. That's the glow we see.
Wow. So the enzyme basically sets up this unstable situation that falls apart and releases light energy.
That's a good way to think about it, yes.
I've heard this described as cold light. What does that mean?
It refers to its incredible efficiency. Almost all, like nearly 100% of the energy released in that chemical reaction comes out as light. Very, very little is wasted as heat.
Unlike, say, an old incandescent light bulb.
Exactly. Those waste a huge amount of energy as heat. Fireflies mastered near-perfect light efficiency millions of years ago. It's something human engineering still struggles to match.
That efficiency is truly astounding. So the firefly has this amazing chemical reaction going, but how does it control it to make those specific flashes, those patterns we talked about?
Control. That primarily involves the nervous system. The current thinking is that they control the flashes by controlling the oxygen supply to the lantern.
So turn the oxygen on, light comes on, turn it off, light goes off.
Essentially, yes. When oxygen gets to the photocytes, the reaction happens, light is produced. When the oxygen supply is cut off, the reaction stops, the light goes out.
How do they manage that oxygen supply so precisely? It seems like it would need to be very fast for flashing.
It does. And research suggests a molecule called nitric oxide might be the key regulator.
Nitric oxide, isn't that important in humans too?
It is, yes. The idea is that the firefly's nervous system releases nitric oxide near the photocytes. This nitric oxide seems to interact with the mitochondria, the energy factories in the cells, and somehow this interaction allows oxygen to flood into the reaction site, fueling the light production. When the nitric oxide release stops, the oxygen flow is restricted again, and the flash ends.
Wow, so nitric oxide acts like a gatekeeper for the oxygen.
That's the leading hypothesis.
Yeah.
But it's important to stress the exact biochemical details are still being worked out. It's complex.
It certainly sounds like it. More than just flipping a switch. You mentioned the unique flash patterns earlier being vital for mating. Can you expand on that?
Absolutely. Those patterns are critical. They're the species' signature tune, if you like. They ensure that males attract females of their own species and avoid confusion with others flying around in the same area.
This prevents accidental interspecies attempts.
Exactly. It maintains reproductive isolation. It's a language of light, and each species speaks its own dialect.
And sometimes you see huge numbers flashing together, right? Synchronized flashing. How does that happen?
Synchronization. That's another really amazing aspect. It's most famous in parts of Southeast Asia. With species like Pteroptics, you can see whole mangrove trees just pulsing with light, all in unison.
That must be incredible to witness.
It is. We also see it in the U.S., for example, in Elkmont, Tennessee, or Congaree National Park in South Carolina, with a species called Photinus carolinus.
Why do they do it? Why synchronize?
Well, the exact reasons are still debated, still being researched, but the main ideas revolve around mating again. One thought is that a massive coordinated flash by thousands of males makes the group far more visible, like a giant beacon, making it easier for females to find them from a distance.
Strength in numbers, visually speaking.
Right. Another idea is predator confusion. A sudden, massive, synchronized burst of light might startle or disorient predators. It could be a mix of factors.
And how do they manage to coordinate? Are they watching each other?
That seems to be the key. The leading theory involves visual feedback. An individual firefly sees its neighbor's flash and adjusts its own internal rhythm, its own timing to match up. It's like they're constantly resetting their internal clocks based on what they see around them.
A collective emergent rhythm. Fascinating. Now, fireflies are amazing, but they aren't the only things that glow, are they?
Not at all. Bioluminescence has actually evolved many, many times independently across the tree of life. It's surprisingly widespread, especially in the oceans. Well, jellyfish, for instance. Acoria victoria is famous, though its green fluorescent protein, GFP, works a bit differently. It's often associated with bioluminescence research. Many deep-sea fish use it, like the anglerfish, with its glowing lure to attract prey.
Ah, yes, the anglerfish. Creepy but cool.
Definitely. And on land, besides fireflies and glowworms, which are often beetle larvae, you have some fungi that glow, like the honey mushroom, Armillaria mellia, or Pinellus stypticus. Their glow is usually much fainter, a sort of eerie green light in the woods.
So they all evolved this ability separately, for different reasons.
Largely, yes. The anglerfish uses it for predation. Some deep sea shrimp use it to startle predators. Fungi might use it to attract insects to spread their spores. While fireflies are heavily focused on mating signals, other organisms show this huge range of ecological uses for self-made light.
It really highlights how useful producing light must be in certain environments. Let's shift gears a bit. I understand scientists have actually harnessed the firefly system, particularly that luciferase enzyme, for research.
Oh, absolutely. The luciferase gene, the piece of DNA that tells a cell how to make the luciferase enzyme, has become an incredibly powerful tool in biology and medicine.
How so? What do they use it for?
One of the most common uses is as a reporter gene.
A reporter gene, like reporting the news?
Sort of.
Mm-hmm.
Imagine you want to know when a specific gene, say gene X, is active in a cell. You can take the controls to switch the promoter region of gene X and attach it to the luciferase gene. Then you put this whole construct into cells.
Okay.
Now, whenever the cell turns on gene X, it also turns on the luciferase gene. The cell makes luciferase, and if you give it luciferin, it will produce light.
Ah, so the amount of light tells you how active gene X is.
Precisely. The brighter the light, the more active the gene. It gives scientists a visual, or at least easily measurable, way to track gene activity under different conditions, like when testing a new drug.
That's very clever. A tiny biological light bulb linked to another gene.
Exactly. It's incredibly versatile.
What are some specific examples of where this is used?
Well, it's huge in medical research. For example, remember ATP is needed for the reaction.
Yes, the cell's energy currency.
Right. So you can use luciferase to measure ATP levels. If cells are alive and metabolically active, they have ATP, and they'll make light with luciferase and luciferin. If they're dead or dying, ATP levels drop and the light dims or goes out.
So you can test if a drug is killing cancer cells, for example.
Exactly. It's widely used in drug screening to test cell viability.
Yeah.
It's also fundamental for bioluminescent imaging, or BLI.
Bioluminescent imaging.
Oh, yeah. You can put cells engineered to express luciferase into a living animal, like a mouse. Then, using a sensitive camera, you can actually see where those cells are and track them over time by detecting the light they emit.
You can see glowing cells inside a living mouse.
Yes, it's non-invasive. It's been revolutionary for things like tracking cancer tumor growth and spread, or seeing how immune cells move around the body.
Wow, that is like having a tiny internal flashlight to watch biology happen in real time.
It really is. And it's all thanks to borrowing the Firefly's light-making machinery. It's also being used to develop highly sensitive biosensors to detect other specific molecules inside cells.
So this beautiful natural display is also a powerhouse for scientific discovery.
Absolutely. And it all comes back to that incredible efficiency we talked about.
Yep.
Nearly 100% conversion of chemical energy to light. Nature's engineering at its finest.
And you mentioned some fireflies can make different colors, not just yellowish green.
That's right. While yellowish green is most common, some species can produce light that's greener, or even orange or reddish.
How do they do that? Different chemicals.
Not necessarily different starting chemicals. It seems to be influenced by the local environment right around the light-emitting molecule, oxyluciferin, at the moment it releases the photon. Things like the pH, the acidity can shift the color.
Oh,
And there's even research suggesting that the luciferase enzyme itself can subtly change the shape or environment of the oxyluciferin, tweaking the energy released and thus the color. It might be that one core molecule, oxyluciferin in slightly different states, can produce the whole range of colors we see.
so the enzyme isn't just making the reaction happen, it's fine-tuning the output.
It seems that way. Our understanding keeps getting more nuanced.
Let's circle back to the control aspect, the flashing. We talked about oxygen and nitric oxide. Can you give a bit more detail on the physiology behind that?
Sure. So that precise timing requires rapid control of oxygen getting to the photocytes. The firefly's respiratory system uses tiny tubes called tracheoles that branch throughout the body, delivering oxygen directly to tissues.
Okay.
In the lantern, these tracheoles end very close to the photocytes. The idea, as we touched on, is that nerve cells release nitric oxide near these endings.
Right, the gatekeeper molecule.
Exactly. The nitric oxide is thought to temporarily inhibit the mitochondria in the cells right at the end of the tracheoles. Mitochondria normally consume a lot of oxygen.
Okay.
So if you briefly pause the mitochondria's oxygen use with nitric oxide, more oxygen can diffuse past them and reach the photocytes to fuel the luciferase reaction. When the nitric oxide pulse ends, the mitochondria start consuming oxygen again, the flow to the photocytes drops, and the flash stops.
Wow, that's a really intricate mechanism, using one cell component to gate the fuel for another reaction.
It's a very clever biological solution for rapid on-off switching.
Are other things involved, like daily rhythms or hormones?
Yes, definitely. Circadian rhythms, the internal 24-hour clock, strongly influence when fireflies flash. They typically have peak activity around dusk or dawn, timing their mating displays for when they're most effective visually.
Makes sense.
But this also means light pollution is a huge problem. Artificial lights at night can really mess up these rhythms and interfere with their ability to find mates.
Ah, that's a serious conservation issue then.
It is. And hormones play roles too, especially during development hormones like juvenile hormone and ecdystroids regulate how the lantern forms in the larva and pupa. In adults, other signaling molecules like octopamine might modulate the intensity of the flashes. It's a whole coordinated system.
Neural, respiratory, endocrine, all working together for that light show. Now, the flash patterns themselves, you gave some examples, like double flashes. Can you give a few more to illustrate the diversity?
Sure. So you have the photoreceptor doublets we mentioned. Some photinid species have single relatively short flashes. The synchronized pteroptic species in Asia often have longer, almost pulsing glows. And then there's spectral diversity too. We mentioned color variation. Luciola cruciata in Japan is known for its distinct green flash. And then you have the really unusual railroad worms, Phrixothrix hirtus, which aren't true fireflies, but are related beetles. The females have rows of greenish-yellow lights along their bodies, like train windows,
Okay.
and two bright red lights on their head.
Red light, that's very different.
It is. And the reason for the red light is still a bit of a mystery. But all this variation in timing, duration, and color is part of that species-specific language for finding the right mate in the dark.
And that synchronized flashing, like with pteroptics, the visual feedback idea is the main explanation.
It's the leading hypothesis, yes.
Yeah.
The idea of phase resetting, where seeing a neighbor flash causes a slight adjustment in your own internal oscillator, pulling everyone into sync. But the exact neural mechanisms, the precise rules they follow, and the full evolutionary story behind why it evolved so spectacularly in some species, those are still really active research questions. It's a beautiful puzzle.
It really is. Okay, let's widen the lens again. We've mentioned marine life and fungi briefly. Can you give a few more examples from across the tree of life?
Absolutely. In the ocean, besides fish and jellyfish, you have dinoclagolates, tiny plankton like Noctilucous scintillans. When disturbed, like by a wave or a boat, they flash, creating that amazing milky seas or glowing wave effect.
I've seen pictures of that. Incredible.
It is. And many marine organisms, interestingly, use a different light-producing chemistry based on a molecule called cholanderazine, not luciferin like fireflies use.
Yeah.
Shows that evolution found different ways to solve the light-making problem.
Convergent evolution.
Exactly. Back on land, we mentioned fungi like armillaria. Bacteria are another big group. Vibrio fisheries is a classic example. They live symbiotically inside special light organs in some squid and fish. They only turn on their light when their population density is high enough, a process called quorum sensing.
So they talk to each other chemically to decide when to glow.
Pretty much, yes. And there are even bioluminescent terrestrial invertebrates beyond insects. Some millipedes, like Motixia sequoia in California, glow quite brightly, likely as a warning since they also produce cyanide.
Wow, glowing cyanide millipedes. Nature's wild.
And even some earthworms. Diplocardia longa can secrete a luminous slime when disturbed. So it pops up in many different groups, serving many different ecological roles.
It's truly widespread. Okay, let's dive just a bit deeper into the scientific uses of firefly luciferase. You mentioned reporter genes and imaging. Are there even more advanced applications emerging?
Yes, the field is constantly innovating. Because firefly luciferase is so sensitive, and because mammalian tissues don't naturally produce light, the signal-to-noise ratio is fantastic for imaging. It allows detection of very small numbers of cells or very low levels of gene activity.
Better than fluorescent reporters sometimes.
In many in vivo, in living animal contexts, yes, because you don't need an external light source to excite it, which can cause background autofluorescence in tissues. With luciferase, the light source is internal.
Right. And the ATP assays?
They remain fundamental. Measuring ATP is a proxy for metabolic activity and cell health. It's crucial not just for drug screening, but also for basic research on cell metabolism and in toxicology for assessing how chemicals affect cells. The speed and sensitivity are hard to beat.
Are there any really futuristic applications being explored?
Definitely. People are engineering luciferase to act as biosensors for things other than ATP. Imagine a luciferase that changes its light output, or even its color, depending on the concentration of calcium ions, or a specific metabolite, inside a living cell. that gives you real-time readouts of intracellular dynamics.
Wow, miniature internal sensors.
Exactly. And perhaps one of the most exciting areas is potential therapeutic use, for example, in photodynamic therapy or PDT.
How would luciferase help there? PDT usually needs external light, right?
Traditionally, yes. But the idea is to target luciferase specifically to tumor cells. Then you administer a prodrug, an inactive molecule that the luciferase enzyme can act upon, activating it to become a toxic compound, or perhaps activating a photosensitizer without needing external light.
So the Firefly enzyme itself generates the light or the therapeutic effect right at the tumor site.
That's the goal. It's still in development, but it offers the potential for highly targeted cancer therapy with fewer side effects, using nature's light source to fight disease.
That is truly remarkable. It really brings home how valuable understanding these natural systems can be.
It does, and the influence is even reaching into art and design. People are using bioluminescent bacteria to create living art installations, and bioengineers are working on putting firefly genes into plants.
To make glow-in-the-dark plants?
Potentially, yes. Imagine streetlights that are actually trees or houseplants that provide their own gentle illumination. It's a long way off, perhaps, but it's being explored as a sustainable lighting concept.
That sounds like science fiction, but grounded in real biology, which sadly brings us to the fact that real fireflies are facing problems.
Yes. Unfortunately, many firefly populations around the world are in decline.
Why is that?
Several key factors. Habitat loss is a big one. As we build more houses, roads, and convert land to agriculture, the meadows, forests, and wetlands fireflies need disappear.
Okay.
Pesticide use is another major issue. Insecticides can kill fireflies directly or kill off the snails, slugs, and worms that their larvae prey on.
Right, disrupting the food chain.
And then there's light pollution, which we mentioned. All the artificial lights from cities, roads, even homes can overwhelm their flash signals, making it incredibly difficult for them to find mates. It disrupts that whole light language we talked about.
That seems particularly insidious for an animal that relies so heavily on darkness and light signals.
It really is. It's a serious threat to their reproductive success.
So what can be done? How can we help conserve them?
Well, protecting and restoring their habitats is crucial, preserving natural areas, reducing sprawl. Minimizing pesticide use, especially broad-spectrum ones, is important. And tackling light pollution is key, using shielded lights that direct light downwards, using motion sensors, turning off unnecessary lights, especially during peak firefly season.
Things individuals can do perhaps in their own yards?
Absolutely. Creating firefly-friendly habitats, avoiding pesticides, and reducing outdoor lighting can make a difference. There are also citizen science projects like Firefly Watch, where people can report sightings. This data helps scientists track populations and understand trends.
It's a reminder that these creatures, besides being beautiful and scientifically valuable, are part of ecosystems that need our protection.
Indeed. So as we see today, bioluminescence is just an incredibly fascinating process. From the intricate chemistry in the firefly's lantern, making that near-perfectly efficient cold light, to the complex behaviors like species-specific flashing and mass synchronization, it's a testament to evolution's power.
And the scientific applications are just staggering. Luciferase really has become a cornerstone tool.
It has. It highlights how much we can learn from and utilize the solutions nature has already developed.
So maybe the next time we see those flashes on a summer night, we can appreciate not just the beauty, but the sheer complexity and efficiency packed into that tiny spark.
Exactly. And perhaps think about the challenges they face in our increasingly bright world. It makes you wonder what other bio-inspired technologies might emerge if we continue to study and, crucially, protect these natural light sources.
A really provocative thought to end on. Thank you. This has been an incredibly illuminating deep dive.